Look at a massive elephant grazing on a savanna, or a towering oak tree stretching into the canopy. Now compare them to a microscopic drop of pond water. Despite the extreme differences in scale, all of them are constructed from the exact same basic building block. We first caught a glimpse of this building block in 1665, when English scientist Robert Hooke placed a cork slice under a primitive self-designed microscope. Through the lens, Hooke saw that the cork resembled a honeycomb made up of dozens of tiny, separate compartments. Because they looked like the small rooms in a monastery, he called them cells. Hooke's observation laid the groundwork for cell theory. This is the biological rule stating that every living organism is made of cells, and every new cell comes from a pre-existing one. Some organisms, like bacteria, spend their entire existence as just a single cell. Others, like us, require trillions of them. In a multicellular body, trillions of cells adopt highly specific shapes and sizes depending on their job. Take the human nerve cell. Its long, stretching tendrils carry electrical signals across vast distances. The form dictates the function. But this raises a question. If a single microscopic cell is a living unit all on its own, how does it actually survive? It doesn't have a stomach to digest food or lungs to breathe. Instead, it relies on a strict internal division of labor. Every cell is wrapped in a flexible outer layer called a plasma membrane. This acts as a highly selective border control, carefully regulating the environment by deciding exactly what gets in and what stays out. Gases cross this border through a process called diffusion. When cellular waste, like carbon dioxide, builds up to high levels inside the cell, it naturally flows out to the lower concentration area outside. Meanwhile, fresh oxygen flows in. Water follows a similar set of rules. The movement of water molecules across this membrane to balance out concentration levels is called osmosis. This diagram illustrates how environments affect the cell. If the outside liquid is highly concentrated with a solute, like salt, water rushes out and the cell shrinks. If the outside liquid is too watery, water rushes in and the cell swells up. Only in an isotonic environment, where the flow is equal in both directions, does the cell remain stable. Plant cells have a unique advantage here. They possess an extra, rigid outer layer called a cell wall, made mostly of tough cellulose. This allows them to swell up with water without bursting. By tightly controlling its imports and exports, the membrane maintains the stable internal conditions required to keep the rest of the cellular machinery running. Deep inside this protected environment is the nucleus. Think of this as the command center. It holds the DNA and chromosomes, the instructions for the cell's design and function. Complex cells, like the ones found in plants, animals, and fungi, are called eukaryotes. They keep their genetic material safely locked away inside a double-layered nuclear membrane. But primitive cells, like bacteria, operate differently. They are prokaryotes. They lack this protective membrane entirely, leaving their genetic material floating in a poorly defined central cluster. The intricate internal structures of eukaryotic cells were completely invisible until the 1940s, when the electron microscope finally allowed researchers to peer inside. What they discovered was an entire microscopic city. While the nucleus holds the blueprints, it relies on a sprawling, highly organized industrial complex outside its walls to actually build the organism. Surrounding the nucleus is the endoplasmic reticulum, or ER. This is the manufacturing floor. The rough ER is studded with tiny particles called ribosomes that manufacture proteins, while the smooth ER synthesizes the fats and lipids needed for the cell to function. Once those materials are made, they are sent off to the Golgi apparatus. This acts as the shipping department, modifying, packaging, and dispatching the products to their final destinations. This transit is constant. Nutrients and proteins are sealed inside tiny fluid sacs called vesicles, which float seamlessly between these factory organelles. None of this manufacturing happens without power. That's provided by the mitochondria, the powerhouses of the cell. They generate ATP, which acts as the chemical energy currency required to run the entire operation. Plant cells have an additional energy source, specialized organelles called plastids. The green ones, chloroplasts, contain chlorophyll and capture sunlight to perform photosynthesis. To manage byproducts of all this industry, Lysosomes act as waste disposal units, using powerful digestive enzymes to break down old parts and foreign debris. And massive storage tanks called vacuoles stockpile excess water, amino acids, and nutrients for later use. Through this precise division of labor, manufacturing, shipping, powering, and cleaning, the cell operates as a fully self-sufficient microscopic metropolis. To facilitate growth, repair injuries, and reproduce, the cell replicates its metropolis through division. For general growth and tissue repair, cells use mitosis. A mother cell duplicates its DNA and splits cleanly into two identical daughter cells, each containing the exact same number of chromosomes as the original. Reproduction requires a slightly different process, meiosis. Here, the cell undergoes 2 consecutive divisions to produce 4 new cells, each carrying exactly half the genetic material of the parent. Every breath you take, every scraped knee that heals, every inch you grow, none of it happens by magic. It is the direct result of billions of these microscopic systems dividing, building, and firing exactly on cue. When you zoom out from that single pulsating unit, multiplying it exponentially, the true scale of biology becomes clear. Final takeaway: you are the sum of trillions of organized